Greeting friends, now we are going to study the next reaction of glycolysis where we convert the glucose 6-phosphate into fructose 6-phosphate. As usual, there are some key points, specific points for this reaction, for this specific reaction. That is, the first is the enzyme. What is the name of the enzyme? That is the phosphoglucoisomerase or phosphohexoisomerase, because glucose is also hexose sugar. Phosphohexoisomerase, phosphoglucoisomerase, both are the same things. This is isomerase, the enzyme that isomerizes these structures. In this case, this is isomerizing this glucose molecule into fructose sugar. The second point is reaction type. This is isomerization, same thing. This is aldose sugar, this is keto sugar. Okay, this enzyme is isomerizing the aldose sugar into keto sugar. That means there is some difference. These are though isomers, that is, they are same forms of this the compound but differing in the stereochemistry, that is the arrangement of atoms. Okay, so they are differing in their stereochemistry. You can see here, this is also 6 carbon sugar, this is also 6 carbon sugar, but the difference is this is aldose sugar and this is keto sugar. The aldehyde group, that is the carbonyl group, is present on the first carbon atom. If we break this structure into the open chain structure, you can see that clearly. And in this case, the carbonyl group is present on the second carbon. Okay, so this is the difference. And the nature of this enzyme, this reaction is that this is, I have not drawn that, this is reversible. Okay, and this is this reaction also needs Mg2 positive for the enzyme to carry out its function. Now there is a question, why a need to convert glucose 6-phosphate to fructose 6-phosphate? Why can't we go on with only glucose 6-phosphate? Why we need to convert to isomerize this glucose to fructose 6-phosphate? Why there is a need? There is a need because there is a carbonyl group. If we break this into the open chain structure, what we are going to see? We are going to see that this is the carbonyl carbon, okay, and this on the left side, okay. If we see clearly, this is the carbonyl carbon present on the first carbon, this is the, and this is second, okay. The point to note here is that we cannot phosphorylate carbonyl carbon. We can't phosphorylate it. Phosphorylate carbonyl carbon. So we need to shift it, and that shifting can be possible when it becomes a keto sugar. When this becomes here, now this group is here now. When it would be in the form of keto sugar, that carbonyl group is in between the chain, and we can easily phosphorylate this thing now, like we did it in the first reaction where we phosphorylated the 6th carbon containing hydroxyl group. Okay, and also if we notice it clearly, then we can find out that this particular figure in the full form would serve as a perfect raw material to convert this into the 3-molecule glyceraldehyde 3-phosphate. and we are going to convert that form into two molecules, that is the 2-glyceraldehyde 3-phosphate molecules would be formed. And for that to happen, we need to convert this into fructose 6-phosphate. Now we are going to study the reaction mechanism of the second reaction, that is the conversion of glucose 6-phosphate to fructose 6-phosphate. Although there is no need, nobody is going to ask the detail of this mechanism in your exams, but you must understand the basic chemistry behind this, so that you are able to understand the meaning of these structures, because you need to study it thoroughly to understand the concepts and grasp all the knowledge behind this. So this is the mechanism of the second reaction. This is the example of acid-base catalysis. So this is acid-base catalysis. It means in all of these reactions, you are going to see an acid is giving its proton, and then the base is abstracting that proton. Okay, so one case acid is donating its proton and the base is abstracting that proton. This is divided into 4 or 5 steps. The first step, in which we are going to see that this is the ring structure of glucose 6-phosphate, and we are going to cleave this from here. So this is going to be cleaved to form it into the open chain structure, and that is done by when the substrate binds to the enzyme, and the enzymatic lysine 518 or histidine 388B, that is the position of these amino acids which are present on the enzyme, they are going to act as enzymatic acid that would catalyze the opening of this ring, and you can see here. Then we can see conserved glutamate 518 that is present on the enzyme would act as the abstracts the acidic proton, and it would act as base. So it is going to abstract the proton from here at the second carbon. We are only interested in this second carbon because we want to convert it in the keto form, and the keto form in case of fructose, it is present on the second carbon. And then the second step is that after when it abstracts the proton from here, this bond will go here, and it will form negative charge, and in the process it would get an H positive ion to make it OH. Here you can see, and then after abstracting the proton, it gives back the proton again. You can see here, this is the proton it abstracted, and now it is donating the proton to make it single bond. You can see here, this is the single bond resulting from the donating back the proton, and from here this is coming here, and we are also removing one proton from here like this, and the double bond position is forming here. Okay, so now you are going to see the keto form here. So this is the keto form. Keto form we have got. Now we can easily phosphorylate this position, first carbon, which we initially couldn't do because carbonyl group was present on the first carbon. Now we have shifted this carbonyl position here in the form of keto group, because when we shift carbonyl position from first to the in between in the chain, then the resulting is the keto form. Okay, so this is the keto form now, and now we are going to close the ring, and that is done by again lysine 518 or histidine 388B abstract back the proton, acting as base, and closes the ring. So this was the mechanism. Thank you.